Hey y'all, and welcome to lecture. This recording will replace our usual live lecture for Tuesday. Today is July 27th, and we are going to cover chapters 12 and 13. This has all already been posted on the module for this week, so please have your PowerPoint and your handout ready to go. And we're just going to jump right in. So today we're going to talk about oral health, risk factors for disease, and a balance that we strike between health and disease in the oral cavity. So what is oral health? And, at first glance, this seems pretty obvious. We've talked about healthy gingival descriptions. We've talked about healthy pocket depths, things like that. But there's always more to it, right? There's always something going on behind the scenes that we don't quite know about yet, or things that we haven't realized indicate health or disease. So we're going to look at these. Oral health is multifaceted. This is not one thing that tells us that the patient's mouth is healthy. So here we have the ability to speak, to smile, to taste food and drink, to touch, to chew, to swallow, and convey a range of emotions through facial expressions. Oral health has everything to do with all of these. And you might think, emotions through facial expressions. Just think if a patient loses their teeth. What is the first thing that happens? They start to lose bone. So their lips look sunken, right? Their face looks smaller. A lot of facial changes. Now, oral health should never include pain, discomfort, or disease. And sometimes disease brings on pain and discomfort. But as you and I know with periodontitis, this isn't always the case. It's usually not the case. So patients don't seek treatment due to the fact that they're not in pain. They think they don't need any treatment. So oral health is a fundamental component of health and physical well-being. If any of you have ever chipped a tooth, lost a tooth, anything like that, you know how this affects us. We feel self-conscious about our smile, especially in the dental world, right? So just imagine if somebody at, 19 lost their teeth due to a drug addiction like methamphetamine use. It's going to impact their physical and mental well-being for the rest of their lives. This is influenced by the patient's values and attitudes, as well as community attitudes and values. What we're seeing currently is a shift toward preventive dentistry versus restorative dentistry. And that's where you and I come in. We're really fundamental to these changes. The next thing, this is influenced by experiences, perceptions, expectations, and a patient's ability to adapt. If a patient loses teeth and they can't eat anymore, that's definitely going to impact their experience and perception of dental care. If a patient had a negative experience or, extremely negative expectations for a dental appointment, that's going to influence their oral health. So there's a lot more that goes into why or why not patients seek help. And this is just the tip of the iceberg. So risk factors, I've shown y'all this picture before. I love this picture. I love it, love it, love it. You can really see how thick that calculus and plaque biofilm is on this extracted tooth. So you can see here, if we know that the cause of periodontal disease, the major etiology is plaque biofilm, that calculus is holding on to quite a bit of plaque biofilm, right? So we have a problem. This is a great way to just look at the different risk factors for periodontitis specifically. Social atmosphere, what kind of attitude was there toward dental care in the patient's home life as they grew up? What's their culture? Do they believe in replacing missing teeth? Do they believe in extracting teeth that are no longer vital? Patients having dental insurance. A lot of patients don't have insurance. They pay out of pocket or they have a very basic plan. Patients' habits. We know a lot of adults actually have no idea how to brush their teeth. And it sounds crazy. How could people not know? But they really don't. They haven't been exposed to the care that they need in order to learn those things. Periodontal pathogens, we've talked a little bit about before. Remember Porphyromonas gingivalis? That's P. gingivalis, also known as Mr. Perio. And then finally, we have systemic conditions. So hereditary, we have diseases like diabetes, and then medications. So let's talk about periodontal disease. This is a multifactorial bacterial infection of the periodontium. Bacteria are the primary etiologic agents in the initiation of disease. Can other things lend to the progression of the disease? Absolutely. But we know that bacteria usually start that process. And the bacteria is harbored in the plaque biofilm, which we're going to talk about a lot in chapter 13. So what does multifactorial etiology mean? Basically, this is the result of a lot of different factors, a lot of different causes. This is why patients have different reactions to the amount of plaque biofilm present in their mouth. Some patients can have super heavy plaque biofilm and be totally fine, hardly have any bleeding, no pocketing. But then other patients have a relatively small or less amount of plaque biofilm and have full-blown periodontitis quickly. So we know that there are a lot of different things that go into the diagnosis and treatment of periodontal diseases. Risk factors are variables that increase the likelihood of periodontitis developing in an individual. So these risk factors don't mean that the patient will absolutely get periodontitis at some point. It only means that they're likely to experience and develop periodontal diseases. So we have a couple different ones here, modifiable and non-modifiable. So I'm going to stick with this slide here. The number one modifiable risk factor in periodontal disease, smoking. So when we say modifiable, that means something that we can change, something that you have control over, that you can change to increase the chance of a positive outcome for a disease. So this is where if we can modify the behavior of smoking and using tobacco, the risk factor of tobacco goes out the window, not a problem anymore. Now, non-modifiable, these are things that we don't have control over. The patient has zero control over. Systemic diseases, if they cannot be controlled, this is genetics, just a myriad of things can cause non-modifiable risk factors. So let's talk about these one by one. Local risk factors are either acquired or anatomical. And as you can imagine, anatomical risk factors, these are part of the anatomy. The patient didn't do anything to have these risk factors, occur. They're just part of the anatomy. Whereas acquired local risk factors, that is caused by something. There is a separate cause for this. It's not part of the patient's normal natural anatomy. Now we're talking about calculus or overhanging margins, poorly contoured crowns. This is where I want you to take a second and think about what all of these things have in common. Calculus is always covered in, overhanging margins always retain, and poorly contoured crowns retain plaque biofilm. So these are local risk factors for the development of periodontal disease, and they are acquired. They're not naturally occurring. We have acquired them through, the means of not getting a cleaning or a poor restoration. Now, anatomical local factors, this has to do with malpositioned teeth. So what I really want y'all to understand here is that we don't recommend braces and orthodontia just for aesthetic reasons. We also recommend ortho for patients with malpositioning to even out that occlusal load on the mouth, to remedy any situations that could retain plaque and therefore lead to periodontal disease. Root grooves. Remember that number 7 and 10, those maxillary lateral incisors have developmental grooves on the lingual surfaces that often cause issues with periodontitis. We have concavities like the mesial concavity of the maxillary first premolar. Those would definitely collect more plaque and harbor bacteria subgingivally that you and I could not get to clean. And then, of course, furcations. So when we think about furcations, it's really difficult for you and I to clean in a furcation area. Next, we have systemic risk factors. Again, this is a systemic disease or a condition. Uncontrolled diabetes. Now, a lot of times I get questions about, well, is diabetes modifiable or non-modifiable? And really that depends. Sometimes no matter what patients do with type 2 diabetes, they still have it, even if they fix their diet and their exercise. In that case, it's not modifiable anymore. Type 1 diabetes is not modifiable. The patient has an autoimmune disorder that's not going to go away. Like I said earlier, tobacco use is the most significant risk factor for periodontitis, and it is modifiable. There's so much that goes into smoking and tobacco use that it gets its own chapter. So we're going to talk about it a little bit later. Medications, you already know, you have to know these 3 medications that cause gingival overgrowth or hyperplasia. The main thing here is that due to the gingival overgrowth, the patient can no longer keep those areas clean at home. Remember that hyperplasia causes deeper pockets without apical migration of the junctional epithelium. So we can't expect patients to clean 4, 5, 6 millimeter pockets at home because there's nothing that can reach into those 7 millimeter pockets, right? And then other hormonal changes like we see in pregnancy or puberty or menopause, stress can be a big risk factor for periodontal disease. We've talked about necrotizing diseases and how it's kind of opportunistic in that the bacteria proliferate when the immune system is down as a result of stress. So now we're going to talk about the balance between periodontal health and disease. So there is, if you think of a scale, and on this side you have health, and this side you have disease. That's what we're kind of juggling here. That's what we're going to talk about. So biologic equilibrium, biologic, study of life, equilibrium, equal. This is a state of balance in the body. It's also known as homeostasis, which a lot of our body processes are aimed at homeostasis, at maintaining the body. So we always want to be in equilibrium. Now, periodontal health and periodontal disease. As you think of this scale, in health, there's a state of balance between biofilm and the host. So bacteria and the host. Remember, bacteria are suspended in biofilm. So if the host is regulating the bacteria enough that there's a balance, they can coexist without disease being present. So anytime we have a bacterial challenge, this has to be contained and tolerated by the host. The host response, which we'll get to probably next week or so, the host response is responsible for keeping that balance. So this is just a fancy way of saying that if the host response can handle the amount of bacteria present, then the body remains in homeostasis. We have a nice balanced equilibrium. In periodontal disease, we see intermittent patterns of disease activity, which means sometimes it's active, sometimes it's dormant or in remission. And the reason for that is because the bacterial challenge is no longer being contained or tolerated by the host. Now the bacteria has taken over and it's heavier, right? So we've lost health. The bacteria side is winning. There are a lot of factors influencing this type of balance. It can be the amount of plaque biofilm, it can be an immune, an immune condition, lots of different things. But at the end of the day, the important thing to take away is that we want to achieve and maintain equilibrium in the oral cavity. And how do we do that? By giving regular preventative dental care. We want to focus on, prophies or perio maintenance appointments and get patients the appropriate treatment right away. So let's talk about that plaque biofilm, which we're going to go into in a lot of detail next chapter. The equilibrium. So some patients, like we said earlier, can go for years and years and years with heavy plaque biofilm. And they might just have gingivitis that never progresses into periodontitis. That means that their host response or their immune system is just handling that bacteria like a champ, not taking any guff from that bacteria, right? Now, some patients progress to periodontitis way quicker than others. So self-care does contribute to this. However, their host response is the major player here, the major player that we're talking about in this balance between health and the biofilm. So other local contributing factors, we have to eliminate the local risk factors. This is going to improve our prognosis. It's going to improve our hypothesis of how the patient is going to heal and how, what's the chance that we can get them back to gingival health, even on a reduced periodontium. If those risk factors cannot be eliminated, and remember we were talking about overhanging restoration, poorly contoured crowns, anything that's retaining plaque biofilm, those local risk factors, if they cannot be eliminated, we have to increase their recare interval. So instead of 6 months, we'll go to 4 months or even 3 months. We double down on patient education. We have to help the patient understand if you don't do these things, you may lose your teeth. So better adjunct aids. If the patient can't use traditional string floss due to dexterity issues, then we need to find something else for them. We need to help them with a floss handle. And of course, explanation of consequences if the patient is non-compliant, which usually patients are non-compliant. So we have to, this is part of when we talked about getting informed consent. If the patient does not follow through with the recommended treatment and the upkeep at home, you have to let them know what the potential consequences could be. In the case of periodontal disease, that means losing their teeth to periodontitis. So systemic and genetic contributing factors, diabetes is a big one. This has so much to do with periodontitis. We haven't established a causal relationship, but we know that they directly influence each other. So diabetes requires collaboration with the primary care physician. There are also some other professionals we should involve here, a dietitian, potentially an endocrinologist, different, doctors with specialties that this patient will have to see. And then smokers, they always require tobacco cessation. So always, always, always go over tobacco cessation. And you're going to say, but Miss Mitchell, What if my patient says they don't want to quit and they don't want me to talk about it? Cool. I'm going to tell you it's your job. They can ask you not to, but you have to tell them, this is my job, and I wouldn't be doing my job if I didn't talk to you about this. You don't have to take my advice, but I still need to tell you what the potential risks are. And again, if we cannot eliminate these factors, then we have to assist the patient by increasing what you and I do. So again, seeing them every 3 months instead of every 6 months. So how do we conduct our risk assessment? Believe it or not, y'all are already doing this in your head. You may not be thinking about it or talking about it out loud, but anytime you do a periodontal chart, calculus detection, periodontal evaluation with a gingival description, when you look at their systemic diseases, those are all parts of a risk assessment, and you're already doing that. So, a risk assessment is basically just identifying the risk factors that increase the probability of a disease. We have individual risk factors, and those involve, again, systemic disease, genetic information, habits. Is the risk factor putting them at a high or low chance of, progressing into periodontal disease? Disease prevention, again, early tobacco cessation and frequent tobacco cessation. Cessation just means stop. So tobacco cessation is to quit using tobacco. If a patient is at risk for developing periodontal disease, we have to address that as soon as possible. And then again, demographics play a big role in whether or not a patient has periodontal disease. And when I say demographics, I mean everything: ethnicity, race, culture, location. Are they out in the middle of nowhere, or are they in an inner city area? What's their transportation like to the dental office? All of these things go into a risk assessment. Medical history, dental history, comprehensive periodontal clinical exams. This is what you and I are doing day to day. This is what we're looking for patients. So here are some different types of risks that can be associated with periodontal disease. We have demographic data like age, duration of exposure to risk factors, self-care. Is the patient taking care of their teeth like they're supposed to? Males have higher rates of periodontitis. Dental awareness, socioeconomic status. Are they aware that they need care? And if they are, can they afford dental care? Medical history, again, tobacco use, diabetes, osteoporosis, which is loss of the density of the bone. This can affect the teeth, specifically the maxilla and mandible. HIV/AIDS is an autoimmune disease, a sexually contracted disease. Genetic predisposition to aggressive diseases. Usually this involves malfunction of white blood cells. So when a patient tells you, oh, I know I have periodontal disease, my dad had it, my grandpa had it, it's not about that. This is genetic predisposition, involves some sort of alteration in the immune system or the immune cells. Dental history, how often are patients getting care? Familial history, how important is it in their family that they receive this care? And then any previous history of perio. So during our clinical exam, y'all already know what we're doing during our clinical exam, so I'm not going to go over that again for you. The big thing here is plaque retention. What plaque retention factors are we looking at? Okay, so let's take a quick break and we'll come back and do chapter 13. All right, y'all, we are back with chapter 13, oral biofilms. This is a really, really important chapter. So if you have any questions while we are going through this, please email me right away. I am happy to help however I can, because oral biofilms are basically the medium in which bacteria are stored and kept against our tissues that cause disease. So in this picture, this is a way you can see, at the beginning, we just got a little bit, then they start to colonize and more bacteria join. Eventually, we've got these like mushroom clouds. And then in step 5 here, this is where the bacteria is free floating again. It lands somewhere else. Process begins again from the beginning. So first, we're going to talk about microbial biofilms, then structure and colonization, and finally, the role of bacteria in periodontal disease. So here's another really great picture. You can see the different shapes here. So we've got I keep trying to draw on my computer screen. Sorry, guys. We've got some cauci, we've got some rods there. There might be some spirochetes throughout here, but you can definitely see the cauci and the rods really easily. So we know that there are up to 1,000 unique bacterial species residing in the oral cavity. Blech, gross. Basically, these have evolved to survive on the tooth surface or epithelium or mucosa somewhere in the mouth. So bacteria can be anywhere in our mouths. A microbe, this refers basically to any microscopic organism. So bacteria, fungi, protozoa, viruses, all of these things make up the microbes that are present at any given time. But first, let's focus on bacteria. These are the simplest organisms, and there are thousands upon thousands of types of bacteria. There are good and bad bacteria in the body. So good bacteria, those are like our gut flora, our normal flora. Those actually protect us and they work to help, keep that equilibrium we talked about in chapter 12. Bacteria replicate very quickly, which allows for evolution kind of on the fly. We see a lot of evolution for bacteria, basically because they're replicating so fast. They can really adapt to the environment that they're in. Now, the bacterial cell envelope, this is part of the bacteria. It's multi-layered and it protects the microorganism from the external environment. So think of it as like a shell surrounding the bacteria. Then we have gram staining, and gram staining is how we classify bacteria into one of two groups based on the structure of their cell envelope. And we're going to go into this a little more in depth here. So the staining techniques that we have available, this has been on the board exam before. So gram stain is just going to tell if it's gram positive or gram negative. The acid fast staining technique, this is how we identify Mycobacterium tuberculosis, which is the etiology of tuberculosis. And then dark field microscopy. So this is used to identify spirochetes. And again, you could see any one of these three on your board. So it's really important to understand the differences here. So the takeaway, really, you and I are going to focus on gram positive and gram negative. So gram positive bacteria stain purple. So think positive, purple. Gram positive, purple. They have a single membrane, which is really important because when we think about membranes, in order to kill bacteria, we have to get through that membrane. So gram positive bacteria have one single membrane surrounding the organism. And it's kind of like mesh. It's like a really thick mesh layer. We also see that they have peptidoglycans, which basically, are sugars and amino acids that make up the cell wall. So amino acids like proteins, those are really important to the bacterial structure. Now, gram positive is susceptible to penicillin. So gram positive, purple, penicillin. That's the easiest way to remember these. Gram positive stain purple and they're susceptible to penicillin. They don't contain any endotoxins, and we're going to talk about endotoxins in just a minute, but you can pretty much figure that anything with the word toxin in it isn't going to be good for us. So, endotoxins are not coming from gram-positive bacteria. Let's talk about gram-negative. You can tell there's a lot more information about the gram-negative bacteria. So, negative, if you're in the negative, you're in the red. Gram-negative stain red. Now, these have two cell membranes, which is important. These are harder to get rid of. So, the outer membrane of the cell wall has receptors made of proteins, and they basically are shaped like this, and they help each other attach and adhere to each other and to surfaces in the mouth. Now, these endotoxins that we talked about on the last slide, these are released when the cell wall is disturbed. So if we disturb the cell wall of a gram-positive bacteria, all right, it's dead. If we disturb the cell wall of a gram-negative bacteria, it releases endotoxins into the environment. These are very, very potent, and they are very destructive. So they cause a lot of damage in the mouth. So the cell wall is located between the outer membrane and the inner membrane, and this is also composed of peptidoglycan. So remember, that was the proteins and sugars that help to protect the outside of the bacterial cell. So tissue damage occurs, usually it can be a direct process, so that bacteria is causing direct damage, or the bacteria is activating the host response. And when the host response activates, we're seeing that our body is attacking the bacteria. However, it's also attacking our own tissues. So it's kind of the 1-2 punch. You can see why gram-negative are the bad guys. We don't want these. These are not susceptible to penicillin. So this is why prescribing amoxicillin for a periodontal infection doesn't work. These bacteria are not susceptible to penicillin, and we're going to talk more later about why that is. So this is the majority of the bacteria involved in periodontal disease, gram-negative. Now, both gram-positive and negative cell walls contain peptidoglycan, but it is thicker in the gram-positive bacteria. So, just think that the gram-negative, remember, if you're negative, you're in the red, has 2 cell membranes, which makes it more difficult to get through than the gram-positive that only has 1 cell membrane. So bacteria are social creatures, and that sounds weird, but it is true. Bacteria like to talk to each other and build communities and colonies with each other. And they are very complex communities, very, very complex. So these little microbial cities basically have tons and tons and species of microbes. Usually they're very well organized, and they're difficult to disrupt. And this is why plaque biofilm has to be disrupted so often. Microbes exist mainly in the colonies, but remember, sometimes we have these planktonic bacteria. Remember plankton from SpongeBob. So if they're in the colonies, they're attaching to living surfaces or non-living surfaces. So the living surfaces are the tooth root, and the non-living are like prosthetic heart valves or implants. So we have to be aware of what our patient is being exposed to when they are getting implants or other surgeries. Now, planktonic, this just means they're free floating. So they can swim around in the saliva and in the oral cavity and go to wherever they want to cause disease. So all of this is held together in a biofilm. I want you to think of biofilm as like the sticky stuff, right? The plaque biofilm is that sticky stuff. And inside of it, we have a bunch of suspended bacteria. So in a biofilm, we have lots and lots of microbial species present. And this is what really helps those bacteria adhere to surfaces. A polymicrobial, poly means many. So anytime you see the word poly, that just means many. Microbial, microbes, so many microbes. Basically, any more than 2, 3 species would be considered polymicrobial. And we know that the biofilm in the mouth is all polymicrobial. There's tons of bacteria in there. So once we start to disrupt bacteria, we've got protective matrix being secreted by the microbes. So all of this stuff is floating around in the plaque on your teeth. Here's what it can look like in the mouth. That's some nice bacteria growth right there. It's disgusting. Now, you know that like pink ring in your sink if you don't wipe it out for a while? That's a biofilm, and it's covered with bacteria in your sink. It's super gross. So this is why we need to clean. So if we clean our sink surfaces, doesn't it make sense that we would also need to clean the bacteria off of our teeth? Yes. So biofilms are everywhere, everywhere. Slime in fish tanks, slime in the sinks, slime on your toilet, slime on rocks outside. These are all different kinds of biofilm. And they can really exist on any solid surface that is exposed to fluid that has microbes in it. Biofilm can form literally anywhere. It just needs a surface, a little bit of fluid, and it's going to grow. So these thrive in dental units, water and suction lines. This is why we have to use water treatments. Sometimes we have them that attach directly to the unit, or we put a tablet in our water when we fill up our bottle to make sure that we don't have any biofilms growing in our water system. And this is also why we run the suction lines. We have to expose that biofilm to enzyme to inhibit it from growing in the tubes. So here are some really common places. Like we said, a fish tank, rocks in a stream. Now, the contact lens cases. Do y'all know how many bacteria can form in a contact lens case? It is disgusting. Don't Google it. You'll never want to wear contacts again. I know people who have Halloween contacts that they wear once a year on Halloween, and the rest of the year it sits in their contact case. I can tell you that is a recipe for disaster. Artificial hip implants, we're worried about any bacterial aggregation there. And then, of course, the tooth surface. This is what you and I are most concerned with. Bacteria starts to form on the tooth surface. So here's a really good illustration of what it looks like. We've got some sort of free-floating bacteria, and eventually it's going to land and attach. Once it attaches, we start to get this secretion of extracellular matrix, and that's what's keeping these bacteria in a colony together. So then we've got growth, detachment. You can see here we're forming bacteria that have little tails on them, and they become, again, planktonic or freely mobile bacteria that can go onto a new spot, just like this, and attach, start all over again. This is why bacteria really can multiply quickly. It's always growing and maturing. So the free-floating bacteria, so we have Staphylococci, so staph, strep, Pseudomonas, and Escherichia coli. So staph, like a staph infection, strep, like strep throat or strep mutans. We have the Pseudomonas, and then E. coli. E. coli is usually a foodborne illness that can make people really sick. So these all form very fast and can cause a lot of issues. What you and I are mostly concerned with here are the streptococci and the staphylococci, because those are also considered normal flora. Some species of staph and strep are normal. So we do have those floating around all over our bodies. So let's talk about how long it takes for microbial communities to form. Literally within minutes, free floating bacteria attach to a surface. So within minutes of being removed, bacteria from other spots in the mouth are already attaching to a surface. Within 2 to 4 hours, they are already strongly attached. Within 6 to 12 hours, they're producing extracellular protective matrix. And this is what's helping them to resist antiseptics and antibiotics. They have that extra layer of protection that any antimicrobial has to really work at to get through. And then 2 to 4 days, that biofilm is evolved into a fully mature colony, and it is extremely resistant to antibiotics. So these recover from mechanical disruption and are reforming within 24 hours. 24 hours, y'all. That is crazy to me. So if that bacteria can reform within 24 hours, does it start to make sense why we tell patients they have to floss once a day? We have to have that disruption of the biofilm, or that bacteria becomes mature enough that it is going to resist our body's natural defense systems. So when patients tell you, I don't understand why I have to floss, this is why. After colonization, we see additional microbes joining the party. So we have the early colonizers are first, and then we have intermediate and late colonizers joining the party. These biofilms enhance the ability of microbes to withstand the host immune system. So our body can't fight these bacteria effectively because they are covered in biofilm. Microbes in biofilms can actually resist factors that would easily kill those free floating microbes. So if we hop back and we look at this picture, these free floating microbes really aren't that dangerous. They don't cause issues for us. Once they become attached, that's where we've got the problems. So the free floating bacteria are not pathogenic as far as periodontal disease goes. It's when they become attached that we really have to worry about them. So there are 3 protective features of biofilm. First, we have blocking. This basically prevents large molecules like antibodies or immunoglobulins from penetrating into the biofilm matrix. So they kind of just shut out everything that our host response is trying to do in order to protect ourselves from the bacteria. Mutual protection. Bacteria will offer protection for each other. This is where, the normal flora of the mouth, the normal bacteria in the mouth try to protect us, but some bacteria actually help each other proliferate. So when we think about normal flora, the good bacteria in our mouth, one thing that bacteria does is help from an overgrowth of fungus within the mouth, like Candida albicans. So this is why sometimes we see patients who are on antibiotics have an overgrowth of Candida. The antibiotic killed all the bacteria, good and bad. So the candidiasis was allowed to proliferate and overgrow within the mouth. And last, we have hibernation. So some antibiotics are useless during hibernation. The antibiotics usually require a metabolic stage to work. So if a bacteria is not replicating and actually ingesting anything from the environment, and an antibiotic won't kill it. Nothing's going to happen. So oral biofilm. I love this picture. It's on your book. It's all over the place. Oral biofilm. This is basically plaque on your teeth. So just think of tons of ingredients in there. Bacteria. We've got metabolic products, salivary glycoproteins. We have tons of microbial species, all in this protective matrix that has been secreted specifically to protect the bacteria from the surrounding environment. So in health, remember that equilibrium we talked about. The bacteria is in balance with the host response. There is not enough bacteria to, overload the host response. Now, if the biofilm is not disrupted, if we're not moving that biofilm off of the teeth, we start to see the imbalance. This is where pathogenic strains of bacteria come in. They start to dominate the environment, and we just have tons of issues. So let's talk about those resident microbes, which are known as our normal flora. Normal flora. These, include bacteria, fungi, viruses, all different kinds of things float around in our bodies all the time. Commensal bacteria, these are part of our normal flora. They live in the oral cavity, and they cause no issues. So remember, all epithelial-lined surfaces are covered in biofilm. Any wet surface is covered in biofilm. Our entire mouth is covered in biofilm. The important thing is it pathogenic bacteria dominating in the biofilm, or is it still normal? Do we still have that system of checks and balances? So symbiosis, this is also known as living in harmony. This is where there's a mutually beneficial relationship between the body and the bacteria in the oral cavity. The bacteria in our mouth actually help us. The normal flora help us digest food, protect from pathogenic bacteria. They help keep the fungi in check. So there's a lot of different purposes for bacteria in the oral cavity that don't relate to periodontal disease. Now, what we want is for those bacteria to help provide nutrition, to help provide regulation of our immune system and protection for our body. Whereas our host system, our body, produces nutrition and environmental stability for the normal or the normal flora, those good bacteria that we're looking at. So symbiosis, this is where we have balance. We have struck a balance between the bacterial load in the mouth and the host response. Now, dysbiosis, this is where there's an imbalance, either on the body or inside the body. So remember, your skin is also covered in tons of bacteria. And if we get a cut, we're worried about that bacteria getting into our bloodstream. So that's why our skin is our first line of defense against disease. So again, if the oral biofilm is not disrupted and it's allowed to mature, then the condition in the mouth is going to start to favor the pathogenic bacteria, which leads to gingival inflammation. So if we're not disrupting plaque biofilm in the mouth, the bacteria become more pathogenic as we let that colony mature. So biofilm bacteria, they're resistant to antibiotics and antimicrobials. So antibiotics like amoxicillin, antimicrobials like Crest or Colgate mouth rinse. Now, this is a crazy fact. Antibiotic doses used to kill free-floating bacteria have to be increased by 1,500 times in order to kill plaque biofilm bacteria. So remember, we said those free-floating bacteria aren't that bad. We can kill those with an antibiotic, no problem. Well, it would take 1,500 times the dose to kill bacteria inside of a biofilm. That dosage would kill a patient before it killed the bacteria in a biofilm. So we're not going to be using that to treat patients who have mature plaque biofilm. Not going to help. So physical disruption of biofilm is required. I'm going to repeat that because you're going to see that again. Physical disruption of biofilm is required. We have to physically disrupt the plaque biofilm on our teeth by brushing, flossing, using instrumentation, whether it's ultrasonic or hand instrumentation. That's all going to be super important. Mouth rinse is not a sufficient means of disruption. This is why we see kids go to college, their mom's not asking them if they brush their teeth anymore, and all of a sudden they have tons of plaque and bad breath because they stopped brushing and flossing. They thought they could just get away with mouthwash. Not the case. So we've been talking about these bacteria and they sound pretty pathogenic. Are they contagious? The short answer is yes. I don't think contagious is the correct word to use. We would rather use they're transmissible. We can transmit biofilm bacteria from our mouth to someone else's mouth through kissing, through saliva. When parents put a child's pacifier in their mouth and then back into the child's mouth, we're actually transmitting bacteria that child is not accustomed to into their mouth. So for all the parents out there, never put your child's pacifier in your mouth and then into their mouth again. You're introducing bacteria that their host response cannot combat. So the Aggregatibacter actinomyces, that's a big one. And remember, that's involved in aggressive periodontitis, as well as Porphyromonas gingivalis. Run-of-the-mill perio caused by P. gingivalis can actually be transmitted from one patient to another. So can strep mutans. So caries and periodontal disease, while they are not contagious, they are transmissible. We can see this go from one patient to the next. So here are a couple different forms, and I'm going to go ahead and zoom in a little bit here. So here you'll see strep mutans. Streptococci are always round. Cocci are round. And then as we move across, so purple is not as bad, and then we get into pink and red, and those are bad. So you'll see the rods, gram-negative rods like P. gingivalis, those are involved in periodontitis. AA, that's involved in aggressive periodontitis, what used to be called aggressive. Then we've got, let's see, the A. nasolendi, the A. viscus, these are all involved in periodontitis. T. forsythia, that's another run-of-the-mill perio one that you're going to see. And then as we get down to the spirochetes, this is where we get into T. denticola. Again, very involved in periodontal disease. So the species capable of colonizing in the mouth, there are 600 that we know of. 600, that's it. 600 that we know of, right? So there's tons and tons and tons more. And this is what it looks like. So again, remember we had those planktonic bacteria floating around. They found a surface to attach to. Some more joined, they're proliferating. Eventually they disperse. We see like this little bubble is going to burst. More planktonic bacteria are floating around and they go to a new site in the mouth. Here's just another picture. This is the one that I think looks like from Star Wars, one of those AT-ATs. Again, just a really, really great description of the phases and what we're going to see from clean surfaces all the way to mature biofilms. So let's talk about these in phases. Phase 1, film coating. This is so important to understand. The acquired pellicle forms within minutes after a cleaning. We remove the acquired pellicle during a cleaning, but it can reform within minutes. There's salivary glycoproteins, antibodies. This is a natural protection of our enamel from acid. It alters the charge and the energy of the tooth surface, which actually facilitates bacterial adhesion. So while it is built to protect our teeth, it also serves as the attachment apparatus for bacteria in the oral cavity. I want you to think of this like double-sided tape. On the inside, it sticks to the tooth. On the outside, the bacteria stick to it. You can't see it. It's just like a layer of protein that surrounds your enamel. So this pretty much provides a sticky surface that aids in bacteria attaching, but because it's attaching to the acquired pellicle, we can still disrupt that biofilm. Here's another really great picture of what it looks like when bacteria start to attach to the pellicle. So that's phase 2, attachment to the pellicle. Within a few hours, bacteria begin to attach to the surface of the pellicle. Well, if we just did a cleaning, why is there bacteria? It is impossible to eliminate 100% of bacteria in the mouth. So some of that free-floating planktonic bacteria is still in there and finds a new surface to attach to. Now, these little guys, they're called fimbriae, and they're little finger-like projections that enable some bacteria to attach really rapidly. It's like they're floating around and then all of a sudden they're latched together. And this is what they look like. These little fimbriae attach to bacteria that get near to it. And then the flagellum, that's like a tail that whips and helps the bacteria move throughout the mouth. So phase 3, this is where we start to get some chemical signaling going between the bacteria. They send out a signal and they're like, party on number 30. And then other free-floating bacteria come to number 30 because there's a party. And then on number 19, they're like, hey, party on number 19, come on over. More of the free-floating bacteria go to that area. So they can actually move through the fluid in our mouths. This is why when we do scaling and root planing, it's really important to scale the entire mouth as soon as possible. If we wait 2 weeks after we do the left side, all the bacteria from the right side has already gone and rejoined. So we start to see coaggregation. This is just a fancy way of saying that a lot of bacteria will attach to each other if they are similar. So remember that in a mature biofilm, we've got tons and tons of species of bacteria. And what happens is we get that initial attachment. Let's see if I can have a picture. All right, we've got that initial attachment and it grows and proliferates. And what happens is that more and more and more bacteria join and we see layers of plaque, layer upon layer. Well, now if these bottom layers closest to the tooth aren't getting oxygen and the nutrients they need anymore, they're actually calcifying into tartar or calculus. So once we have layer upon layer of plaque, it calcifies and it becomes calculus. And then the calculus is covered in plaque and so on and so forth. This is how we keep forming layer upon layer of biofilm and calculus on the teeth. So once we get, that stacking of bacteria, the different shapes are going to stack on top of each other. Then we've got the matrix making up the biofilm, and now we have a mature colony of bacteria. So co-aggregation is not random. This is a very well put together community of bacteria in the biofilm. So each bacterial strain has a limited set of bacteria that they can adhere to. So they're seeking out specific bacteria that they can adhere to easily. And the ability to do this is really important for bacteria and for the formation of those colonies in biofilms. Now, phase 4, we start to see this slime layer. The extracellular protective matrix is also known as the extracellular slime layer. You might hear it either way. This is secreted by bacteria, and it has proteins, glycolipids, bacterial DNA. It's very, very sticky, and it really helps that plaque biofilm adhere to surfaces, and then the bacteria to adhere to each other and surfaces within that sticky substance. So this slime layer basically is helping to anchor everything and keep it against the tooth and the tissue. So it's protected from antibiotics. It's now protected from the immune response. It's got a nice little colony just hanging out like at a spa, right? And they're all growing and proliferating. Next, we have microcolony formation. The bacteria proliferate, grow away from the tooth surface. We see those blooms. They're kind of like mushroom shaped, and they grow really, really fast. Eventually, we see those really narrow bases, kind of like pedunculated, where it becomes like a mushroom shape. And we have lots of bacteria up in here. And then finally, we get to, well, not quite, I guess, but in stage 4, there's lots of different things going on. So each microcolony is a tiny independent community. It's got thousands of bacteria within it. Then we have different microcolonies that contain different combinations of bacterial species, which means that within one pocket, one gingival pocket, we've got all these different formations of bacteria. And each formation has multiple different types of, species of bacteria in it. So we're starting to see a lot of issues. Let's break these down one by one. So the environments within the microcolonies, it's very unique to each colony. The differences, we've got differences in oxygen concentration. Is it supragingival? Is it subgingival? That's going to impact how much oxygen is available. The pH, what is the pH of the mouth? Is it acidic? Is it basic? Is it neutral? And finally, temperature. We know bacteria like a warm area, right, to proliferate. The mouth is moist and warm, and it's perfect for bacteria. So you've got a little incubator right in your mouth. Each species prefers a certain environment within the biofilm, and that's how we know the difference between bacterial species on the supra and subsurfaces. It ensures the survival of plaque biofilm. So, basically, depending on that patient, the bacteria is able to make a colony that is resistant to everything that we're throwing at it. And if the plaque biofilm, it's multifactorial, there's lots of different species. If it was just one species, we could destroy it easily and it wouldn't live. And that's not the point of bacteria. Bacteria want to proliferate. Now, fluid forces. This is so important. Not only do the fluid forces shape the biofilm, they also result in extensions from the main body of the biofilm. So let's go back to this picture. These extensions here that are kind of reaching over to other colonies, that's a big deal because they can break free. We can swallow them, we can spit them out, or they can go form their own colony in other areas of the mouth. So these fluid forces kind of help with cell to cell collisions, and that's where the bacteria can really spread. And it helps a lot with constant evolution of the bacteria. So we see them become more pathogenic as time goes on because they're communicating with each other. Now, those fluid channels, they can penetrate the extracellular slime layer. So remember, the slime layer is built as a protection, but these fluid channels can get through because they're bringing nutrients and oxygen. Well, bacteria are living organisms. They need nutrients and oxygen to survive. So these fluid channels are just taking it through. They also take out bacterial waste products. So this can be beverages that we drink, it can be saliva, it can be any liquid in the mouth is helping to create these fluid channels. So what is this cell-to-cell communication system? There is direct interaction where they can collide with each other and then adhere, but they also use chemical signals called chemotaxis. Chemotaxis is a chemical signaling system in the body. They can communicate with each other. And this results in the transfer of genes among bacteria. So this one is like, hey, I'm really pathogenic. Here, have some of my DNA, and you can be more pathogenic too. Not great. Not great for us at all. So bacteria within a biofilm, this signal, they've got tons of proteins that the free-floating bacteria don't have. So some of those proteins actually trigger adhesion of bacteria and formation of the extracellular slime layer. So they get into this community, this little colony, and they're like, okay, we're safe now. Let's, secrete our protective layer and protect our house kind of thing. So dispersion, that is a lot of plaque. There's probably food in there. We've got a lot of inflammation on this patient, right? So once this is here, the bacteria can actually detach from the colony. And let's say they were like, hey, this one doesn't have a lot. Let's go to this tooth. And it starts all over again. That bacteria can spread and colonize new areas. It is like Great Britain. They just want to colonize everything. So that bacteria goes from well-developed microcolonies to other areas and starts all over again, which for you and I is not great because we have to get that under control. So here's a really good picture of what we're seeing. Now I'm going to zoom in, just kidding, on this one. Okay, there we go. So this is our enamel. This is our acquired pellicle. And then these are the proteins that extend from the acquired pellicle. What you're seeing is that first, we've got cossi, right? So cossi are always first to colonize. Then we have these rod shapes, and then eventually we get to these spirochetes. So this is what I was talking about with aggregation of bacteria. The early colonizers are the cossi, and then we move forward from there. So early colonizers, these are non-pathogenic. The first bacteria are non-pathogenic. Usually it's normal flora, whatever's floating around in our mouth. Those are the first to colonize. And, basically if they can attach, we've got biofilm coming. If those attach, biofilm is on the way. Next, we've got periodontal pathogens, right? So periodontal pathogens are unable to colonize the biofilm until we get those non-pathogenic species attached. So we have to have those run-of-the-mill Streptococcus attached to the acquired pellicle before we start to get any dangerous pathogenic bacteria into that colony. So Periodontal pathogens are freely floating until early colonizers attach to the tooth. Then they send out those signals and say, hey, come on and join the party. They join the biofilm, and then the conditions are favorable for proliferation of that bacteria, which again will lead to dispersion and recolonization in other areas of the mouth. Next, we have the early colonizer streptococcal species. They attach really well. So this is what we're seeing. This here is the tooth surface. And you can see as we get more and more bacteria in these fluid channels, and here's some fluid channels in here. Look at how close that bacteria is coming to the epithelium. Too close. So the more plaque biofilm we have, the closer it extends out to the epithelium from the tooth surface. And it's going to start causing a lot of issues. So remember that the streptococcal species usually are the ones that attach to the tooth and the pellicle or to each other, but some of them can't attach. There are only some that attach, and then the others co-aggregate on top of those. So here's what it looks like. If this is our tooth over here, first we've got our row of cocci, usually streptococci. Then we've got some rods attaching. Then some more cocci. Then we start to see these corn cobs where it's a rod surrounded by cocci on either side. Then we get these gram-negative, and eventually we start to see spirochetes. So this is what it looks like after one day. This is why patients have to floss at least once a day to disrupt this biofilm. Otherwise, it hardens into calculus. So intermediate and late colonizers, they have to join biofilm in this sequence: early, intermediate, late. A lot of periodontal pathogens are actually late colonizers. Here's another great picture of what it looks like. So these have names in them. I don't need you to memorize the names, but it's important to know we go from gram-positive cocci, gram-positive rods, then to gram-negative cocci and gram-negative rods, and eventually we get over to the spirochetes. So early colonizers, the free-floating periodontal pathogens cannot cause disease. I've said that twice. You know what that means. And basically, we need those non-pathogenic early colonizers before we can expect that pathogenic bacteria to come into the environment. As soon as we disrupt biofilm, it starts reforming. Remember, within minutes, we have the pellicle attached, or the pellicle formed, and then the bacteria start attaching right away. So let's start at the beginning. Early colonizers. These are gram-positive, like Actinomyces viscus and Streptococcus sanguis. So they form this layer around the acquired pellicle. Gram-positive bacteria always colonize first, specifically cocci. Gram-positive cocci colonize the pellicle, and that's where the rest of the coaggregation starts. Now, gram-negative bacteria start to join in. This is where we see Prevotella intermedia. Y'all remember P. intermedia from pregnancy gingivitis? So we've got our gram-positive, then we get into gram-negative cocci and rods. And then finally, we get into the really bad guys, those gram-negative bacteria like P. gingivalis. This is a gram-negative rod. You have to know that. P. gingivalis is a gram-negative rod, and it's going to start causing a lot of issues and co-aggregation of bad bacterial species for the patient's health. So let's talk about that colonization and sequence. Remember, within hours, we've got the pellicle and early colonizers attaching. In 6 hours, the surface of the crown becomes covered in biofilm and bacteria. By day 7, we have mature supragingival. And then between 3 and 12 weeks, we start to see subgingival biofilm. So every single day, we need to disrupt that plaque biofilm. So let's talk about the attachment zones. We basically have the root surface or the epithelial lining of the periodontal pocket. And we know that the tooth surface is where it starts. But as that bacteria co-aggregates and the colony gets larger, it starts to extend out to the epithelium. And we're actually going to see this all the way from the marginal gingiva, the gingival margin, to the junctional epithelium. Bacteria is just going to cover that epithelium. And then those subgingival bacteria have the ability to invade the dentinal tubules. Now we're starting to see some real big problems. The cossi and rods dominate, and we see lots of filamentous, you remember those little filaments that help them attach? That's what we're going to see here. So on the tooth surface, we start to see demineralization. And then on the epithelial surface, this is where we see that bacteria is going to start marching down toward the connective tissue and causing loss of attachment in periodontitis. Now, tissue-associated plaque biofilms. This is where the bacteria adhere to the epithelium, and it's very loosely attached. These are very different from the tooth-associated biofilms. But basically, this is what invades the connective tissues, the gingival tissues, the periodontal tissues, and the surface of the alveolar bone. There can also be free-floating or planktonic bacteria that's not part of the biofilm colony in the pocket, but most likely what's going to happen is that's going to go to another area and colonize by itself, start a whole new colony. So there are lots of spirochetes. Remember, spirochetes are the really bad ones, and then flagellated bacteria, the ones with the flagella that help them move around. So what does this mean? Why is this important? Why did we spend 2 hours talking about it? Biofilm bacteria are extremely resistant to antibiotics, antimicrobials, and the immune system. Remember that extracellular slime layer is its layer of protection. So some bacteria can actually produce enzymes that degrade antibiotics faster than the drug can penetrate into the biofilm, like amoxicillin. Remember, we said we would have to have 1,500 times the dose for bacteria involved in biofilm as compared to free floating. So when we finish antibiotics, any dormant bacteria within the biofilm reactivate. Remember that hibernation protection mechanism? When bacteria sense that there's something coming after them, they can hibernate, and then the antibiotic isn't going to help. So physical disruption of biofilm is required. Always. Brushing, flossing, periodontal instrumentation, whatever we can do to physically disrupt biofilm. Now, a water pick is a great way to disrupt biofilm. However, it still doesn't clean the contact area between two teeth. So it does not replace flossing. However, it is great for bacterial death in the pocket. So let's talk about these different hypotheses involved in the progression of periodontal disease. First, we have the non-specific plaque hypothesis, and this basically said that all plaque biofilm leads to periodontal destruction. Well, you and I know that's not true, right? Why do some people get gingivitis and it never progresses to periodontitis, while other patients have really light plaque biofilm and have crazy periodontitis? It doesn't explain the difference in rates of periodontitis and destruction in some areas of the mouth while there's none in others. Next, we have the specific plaque hypothesis. And this meant that specific bacteria present in biofilm result in periodontal tissue destruction. Basically, gram-negative anaerobes were thought to be the only cause of periodontal diseases. And these were called the Safransky's bacteria. So organization is not random. We talked about it. Cauci, then rods, then spirochetes. Gram-positive, then gram-negative. Aerobes, then anaerobes. So there are specific associations among these species, and Safransky grouped them into complexes. So the colors are based on the association with health or disease. So I want you to think of this as a green light, a yellow light, or a red light. Yellow and green are early colonizers, and they're usually present in health. You can go through a green light, you can go through a yellow light. But then I want you to think of orange as the light right before it hits red. If you go too late through the light, it's almost red. So it's orange here. And these were thought to be the major etiologic agents of periodontal disease, specifically the red complex bacteria. And if you'll think back to when Marie did our Arrestin presentation, she mentioned these bacteria quite a lot. The red complex bacteria, P. gingivalis, T. forsythia, T. denticola. These are thought to be the major bugs or bacteria in perio. And even though this is a little outdated, the board is still covering it this way. So that's why we're still going to learn it this way. Now, some problems here. We have new, better detection methods that have told us that some of those red pathogens can actually be present in health. So they can't absolutely be the cause of periodontal disease if some of them are still present in health. Now, current perspectives, host response. The host response is the primary predictor of prognosis of periodontitis. The host response. So the host response, not the bacteria, determines periodontal disease and destruction. And inflammation is what's driving those changes. And, not that the bacteria are causing more inflammation, but the inflammation is causing more problems than it is solutions. All right, guys, we made it. We made it to the end. Like I said, if you have any questions, please, please, please don't hesitate to reach out. This is a really big chapter. These are not covered on your exam for Thursday. That will only be those chapters. I've already posted your study guide. So if you need anything, please reach out and I will talk to y'all soon. Thank you and have a great night.